Pharmaceutical Processing, in conjunction with INTERPHEX 2016, presents INTERPHEX Live.
Hi, my name is Russ Soma. I'm owner and founder of Somatech LLC. We're a consulting company and we're affiliated with the engineering firm IPS in Blue Bell, Pennsylvania. With me today is a good friend of mine and one of my fellow graduate students about 200 years ago, Bob Timko. Bob Timko is the founder of the Rotar Pharma Consulting Company. And Bob, why don't you tell us a little bit what you do there at Rotar?
Well, Russ, as you, I'm also a consultant. I work in the area of pharmaceutical development and regulatory CMC sciences for a variety of small and upcoming companies.
Well, CMC, that was a great way to leverage all of your background and your pharmaceutics knowledge. That comes in handy when you're putting together those filings, doesn't it, Bob?
It sure does. It makes having some knowledge of that area quite helpful in working with a lot of folks.
Yeah, I'm always amazed when people refer to what we do as CMC stuff. You know, I used to think of it as pharmaceutical technology, but what are you going to do?
Well, that's the simplest way for people to understand it, so we go with that.
Okay. Well, while we're on the subject thereof, one of the things that I think we both have been wrestled with, not only in our former lives as big pharma employees, but as consultants, and that is the enhancement of bioavailability. And nowadays, some of these drugs that are coming out, especially in the oncology area, they have the solubility of sand. So, what are some of the considerations that can be or the hindrances that you've seen in trying to enhance bioavailability?
Well, as you mentioned, Russ, the biggest thing for the drugs today that we've seen coming across is their aqueous solubility. If they can't be dissolved or they can't be soluble in the aqueous media, they have a tough time being absorbed and getting across the membranes of the intestinal guts. So really, it's a combination then of solubility and permeability, and that's sort of outlined in the biopharmaceutical classification system, the BCS system, which addresses permeability and solubility of drugs.
Okay, great. I think let's get to that. I want to hold that BCS till we get a little bit later. Why don't we touch on the physical chemical properties of the drugs? I mean, people always think solubility, but what else? I mean, you and I both know there's a lot more to it than what do you look to?
I mean, when you first take a look at a drug, the one thing you want to understand is a little bit about its physical chemical characteristics, as you mentioned. You want to take a look at whether the drug is a crystalline solid, whether it's amorphous, whether it's a acid or base, a salt. You might want to also take a look at its particle size. All those have an effect on possibly solubility and permeability and the ability to be bioavailable.
I gotcha. So we're talking about crystallinity, we're talking more amorphous, less amorphous. I mean, it becomes intuitive, obviously amorphous seems to be more soluble. But tell you what, why don't we talk about now, what are the technologies to enhance this? We can start, let's talk about particle size. So what technologies can we throw at this to enhance bioavailability?
Well, I mean, it's really, rather than technologies, I think really it's techniques that you might want to consider. One of them, obviously, is whether or not you're gonna, look at whether there's salt formation, whether or not, as we mentioned, whether or not you could reduce the particle size of it, whether or not you could mix it with other materials that enhance its solubility. So you'd want to look at some of those areas first before you'd go into anything really exotic. Look at what the properties of the drug and how it interacts with its general environment is to see best for getting solubility and permeability.
And when we're talking about particle size reduction, Bob, I mean, we're not simply talking about running this thing through your standard co-mill or Fitz mill.
Oh, no. I mean, if you need to look at particle size reduction, sometimes you have to look at things like a jet mill or a nano mill, one of those that reduced particle size down to 2 micron or submicron levels in order to possibly enhance stability. And then when you get down that low, the issue is how do you stabilize it in the best way to ensure that you keep that particle size small enough so that it rapidly dissolves, or at some level dissolves sufficient to be able to get into solution and across the intestinal lumen.
I got you. So, but how about this mixing with other stuff? I mean, this is like co-extrusion. What are we talking about there?
Well, I mean, there's a variety of techniques that you could do there. You could do things like simple solid dispersions, eutectic mixtures. You could do things like melt technology. Solid dispersions where you melt the drug, dissolve it in a medium. You could do spray drying. A number of those technologies are available that may help enhance solubility and processability of the compound.
I got almost as if you wanted to stabilize an amorphous structure.
Pardon me, Russ.
Yeah, I'm thinking perhaps these technologies we throw at it is the attempt to stabilize an amorphous structure to enhance solubility, or?
Well, I mean, there are a number of amorphous compounds are generally less stable than crystalline compounds, and so you may end up having to mix them with some type of polymer system or some other means to stabilize them to enhance their solubility. I mean, there's a number of patents out there where they've mixed them with things like PVP or HPMC to stabilize, an amorphous drug and at the same time enhance its bioavailability.
I got you. So, we're trying to, make some of this stuff available so people say, hey, I was listening to what Bob was talking about, and yeah, I wanted to try that. So I think along the lines of let's try something like that, what are some of the manufacturing techniques or equipment that one would look at? So, we were talking about co-extrusion. So what are we looking at as far as equipment to do this work?
Well, I mean, if you're looking, first of all, you're going to try the basic techniques. You're going to look at some things like jet mills or nano mills in order to reduce particle size. Sometimes it's even as simple as using a hammer mill where you have a fairly large crystalline material and you want to reduce it to a smaller size. It's not all the time where you need to go to an exotic technique, but jet milling, which uses some air, wet milling techniques, which is maybe a nanoparticle sizer, could be used to reduce particle size. Again, a spray dryer where you're mixing the drug with an aqueous media and you may end up producing a stabilized amorphous form. So there's a couple of those technologies that you could look at.
So it looks like the first thing to do is see if you can reduce the particle size as simply as possible, and probably a hammer mill to do something like that. And then if that doesn't work, move on to something that's a milling technique that's probably just a little more intense. And if that doesn't work, start looking at some kind of co-extrusion or mixing or amorphous solid. Is that it? You think that would be the progression?
I would say that those are the techniques that you would basically want to start. You don't want to start with the exotic stuff first, because sometimes the simplest way is the easiest way.
That's it. Well, don't we know that though, right? So, one of the things that, one of the things I think we always have to address here is, and again, especially with your CMC background, is a lot of cases is when they want to go to make the filing or when they want to put this stuff together, quality by design. I mean, the reason is let's cut right to the chase. In a situation like this, the quality by design, the knowledge that you would gather is important. So what sort of knowledge would you be gathering up there so you would be able to build this into your CMC section to enhance, in other words, my objective is to enhance bioavailability. What's the laundry list of information I'm going to collect?
Well, I mean, you really start, quality by design is really about understanding, material, formulation, and process understanding. So you really want to understand the basic compound you're dealing with, its physical chemical characteristics, how that compares to your knowledge base of materials that you may have worked on in the past. Is it more soluble, less soluble? What worked, didn't work? And then taking that information and using it to design how you're going to develop your formulation. What are the risks? What are the critical attributes that you want to address, and go about it with that approach. It's to me, quality by design is really understanding what you want to do, why you're doing it, and understanding what the result may be once you get it, and what's critical and not critical.
That's a good point, Bob. I mean, how far back? I mean, let's just take a page out of your CMC book for a minute. I mean, how far back does the information go? I mean, for example, how about salt selection? I mean,
absolutely. I mean, to me, quality by design goes back into if you're looking at a compound, it goes back into why you chose the form you chose. Whether it's crystalline, whether it's amorphous, whether it's a hydrochloride salt, or whether it's a sulfate salt, or an acetate salt, whatever. It all depends. That's all part of the paradigm, the quality by design paradigm, and understanding what you're doing from a drug discovery, drug development, drug selection process, all the way through to eventually your manufacturing process. That's all part of this whole, as I call it, quality by design paradigm, and the understanding that comes with it.
how do you get a baseline on? I mean, let's assume that you didn't make the classic wrong salt selection and you got the right salt. I mean, how do you get a baseline blood level on stuff like this? I mean, let's assume it's a tough one. What's your recommendation there? What's the classic approach?
Well, I think what you want to do is understand what you've done with some other compounds. Here you may have to look at some animal testing, animal models, look at which salt is more stable, which may give you some better results in some very small populations of animals, and use that, because for many compounds, before you go into the phase 1, phase 2 clinical, you have to do the safety and tox studies. So you can't really do the early bioavailability work on humans. You have to probably do a lot of the work based on what you've seen and what you're able to do in some animal models for solubility and permeability testing, and use some of those techniques from the BCS classification system, where they look at permeability in KCO2 cells, or they look at solubility studies for those aspects.
Yeah, earlier on we said we were going to talk about, I think real quick, one of the things we should say is, I think it's important to characterize the BCS of the drug, and why don't you touch on that a little bit, Bob?
Well, I mean, the BCS system was developed a number of years ago, and what it relies on is solubility and permeability, and they basically break drugs down into four quadrants, as they call it, based on those characteristics. BCS class 1 are high solubility, high permeability drugs. Usually those type of drugs have very little issues with bioavailability and being freely soluble and easily pass through or easily absorbed. Then there's the low solubility, high permeability drugs, where the solubility becomes more of an issue. And I believe that's class 2. And then there's class 3, which is high solubility, low permeability, which is class 3, and then low solubility and low permeability, which is class 4 drugs. So they tend to look at those characteristics.
And I think one of the things too, Bob, I think that we didn't discuss in great detail, but it's the environment, the microenvironment of the dosage form itself, to mitigate dissociation such that you do get permeation. I mean, what's been your experience there in dosage form development?
Well, some of the experience you find out is microenvironment in the dosage form is very helpful in aiding or helping improve solubility or permeability. Many times, for example, if you have a poorly soluble or basic drug, you may be able to use an acidifying type excipient to help solubilize the drug as the dosage form disintegrates and dissolves within the intestine. So it depends really sometimes by what you mix the drug with is important as just as much knowing what its physical chemical characteristics are. It's a combination of that is quite helpful in the formulation prospect.
I think just what we should do for a recap then, let's just go over, okay, you present it with a drug, what's the stepwise process for me to say I'm going to get the best bioavailability I could possibly get out of this compound?
Well, I guess first of all, you start with the physical chemical characteristics. You start out with understanding whether or not it's a solid, crystalline, amorphous material, whether or not you're dealing with a salt, what its pH solubility profile is, what its stability in solution is, what's its optimum stability at pH, what affects it. From that, you may look at particle size of the drug, means of how to reduce the particle size, whether it's by simple mechanical means, whether or not you need to micronize it, wet mill it, whether or not you may need to mix the materials and perform some sort of dispersions or mixtures with them, whether you could adjust the pH of this formulation. It's a series of progression. All that is, and that's all tied up into what this thing we talked about called quality by design and understanding. So it's a concept that starts with the drug compound synthesis and ends with hopefully a commercial dosage form where you understand why and how you got there. And hopefully it all is bioavailability and therapeutically effective.
Well, that's really the end game when we're hoping for is the product quality. Well, Bob, it's been a delight to discuss this aspects with you, and we both know that enhancement of bioavailability is something that we deal with on a regular basis. So, with that, I would say that I think should anybody have any other questions, again, this is Bob Timko, Rotor Pharma, and this is Russ Soma from Somatech LLC. Thank you very much.
Pharmaceutical Processing, in conjunction with INTERPHEX 2016, presents INTERPHEX Live.